Now we come to the last topic of mineral nutrition in plants. Our understanding of entire plant nutrition is incomplete if we don't understand the nitrogen nutrition in plants, because we have realized the importance of nitrogen in various biomolecules which are of great importance. Like for instance, the DNA, that is the nucleic acid, would not be formed if nitrogen is not present. So nitrogen plays a crucial role in various biomolecules which are important for survival of the plant. So we are going to study nitrogen nutrition in plants, and this is the last topic that we have in mineral nutrition. So there are a few questions that we need to answer by the end of this topic, that is that what are the available sources of nitrogen, how this nitrogen is available to the plants. Once it is available, how that nitrogen assimilation would take place. What is basically nitrogen assimilation? What are the types of fixation of the nitrogen, whichever is available to the plant, how that nitrogen is fixed for the plant to be available for the plant, and what is the nitrogen fixation process in case we are talking about leguminous plants. So by the end of this lesson, you must be able to answer all these questions. So first of all, see that what are the available sources of nitrogen. When we are able to answer these questions, we will get our clear-cut understanding of the nitrogen cycle which is taking place on earth. First of all, you know that our atmosphere is almost 70% of the atmosphere is nitrogen. So the available source of nitrogen is atmosphere basically. But to our dismay, you must be knowing it, that atmospheric nitrogen is present in the form of dinitrogen, that is this molecule of nitrogen, and plants cannot take up that nitrogen directly. So the available source of nitrogen would be either the atmospheric nitrogen or the nitrogen which would be present in the soil itself. Now for the plants, the available nitrogen would be in the soil, and in soil it is present in the form of, first thing is nitrates, nitrites and ammonium ions. Okay. So these are the three sources which are available to the plant, and they are present in the soil. So first thing that we had to understand was how it is available. So let it be available in the form of nitrogen gas in atmosphere, and in case of soil it is present in the form of nitrates, nitrites and ammonia. How these nitrates and nitrites are being made available for the plant, that is the type of nitrogen fixation. Alright, this atmospheric nitrogen serves as the source of nitrogen which would form the nitrates, nitrites and ammonia in the soil. So how they are formed? This atmospheric nitrogen is under the agency of few physical processes, like supposedly I am talking about lightning. Okay, we will understand it with a pictorial representation that I am going to show you in some time. So physical processes like lightning, what they do is that they break the nitrogen into nitrates and nitrites. Okay, or the gaseous part of the nitrogen would be converted into acid rain sort of thing, that the nitrogen would be converted into oxides. The oxides would come in contact with water and they would form nitric acid, and that would be available to the plants through the soil. So lightning is an important process. Apart from that, whatever ammonification is taking place, ammonification is the process in which the ammonia is being added to the soil, and that is taking place in the process of decomposition which keeps on taking place in the soil. So the process of decomposition adds ammonia to the soil, and by the agency of microbes like Nitrosomonas and Nitrobacter, the ammonia is first converted, please pay attention, ammonia is first converted to nitrites, which is further converted into nitrates, and from here the plants can take the nitrates directly for the assimilation. Now the question comes, what is nitrogen assimilation? Nitrogen assimilation is that process in which, whether in the form of ammonium ions, whether in the form of nitrites, or whether in the form of nitrates, whatever the plant has taken up, that would be utilized in the formation of amino acids and other biomolecules which the plant requires. That is what is nitrogen assimilation. For that, first of all, the atmospheric nitrogen needs to be converted into these forms. For there, where we have various biological agencies and we have physical agencies. So the two types of nitrogen fixations on the basis of agency for fixing that nitrogen, we have first one being physical nitrogen fixation, in which we discuss lightning and other decomposition processes come into scenario, and the second one is biological. In case of biological nitrogen fixation, there would be enzymes which would be involved, there would be prokaryotic cells which would be involved for the fixation of atmospheric nitrogen into the plant, and for that they need to convert the atmospheric nitrogen into ammonia. Once it has been converted into ammonia, it would be further converted and not converted, basically it would be further undergoing few processes like reductive amination, which we are going to study. But for now you need to understand that the atmospheric nitrogen somehow in the presence of an enzyme would be taken up by the roots of the plant, and then it would be converted into nitrates, or it would be directly used for the processes of formation of amino acids, that is reductive amination. Remember the name, we are coming to that in some time. So in case of biological nitrogen fixation, we have to understand that the biological nitrogen fixation would not be possible without the presence of prokaryotic cells. Now why I say prokaryotic cells? Because the prokaryotic cells on this earth are the cells which are the exclusive, which are with exclusive ability of fixing the nitrogen, that is biological nitrogen fixation. That means they would take up the atmospheric nitrogen and convert it into ammonia for the plant. So we have symbiotic biological nitrogen fixation, in which the prokaryotic cell would be in symbiotic relationship, or in other terms mutual relationship with the plant roots, and we have the example of rhizobium. Whenever we study this symbiotic biological nitrogen fixation, we see in leguminous plants the rhizobium genus is very important and crucial for forming the nodules in which the entire nitrogen fixation takes place. That is why it is said that pulses are very rich source of nitrogen and proteins, because they have legumes in their soil. Not soil, pardon me, it's not the soil, they have legumes in their roots, and those legumes are formed by the bacteria of these genus. Apart from this, there is another genus, that is Frankia, which also forms nodules in the plant, symbiotically associated with the roots, and they help in nitrogen fixation. There are certain bacteria which are not in symbiotic relationship, like Azotobacter. They are going to convert the nitrogen into the ammonia by not getting associated with the plant roots. They would convert that atmospheric nitrogen in the soil itself, either into the form of the nitrates or nitrites, or they would convert it into the form of ammonia. They would use the enzymes like nitrate reductase and nitrite reductase, which would convert the nitrates to nitrites and nitrites to further ammonia, and these are asymbiotic bacteria. We have two examples for each. These do not form any nodules. Those which form nodules, we are going to study those bacteria basically. So what happens in the biological nitrogen fixation in the case of rhizobium? Now you know, a plant is having its roots, it is having its rhizosphere. So you come across, I am just giving you a vague idea of a root. Let this be the primary root which is going down, and these are the secondary roots, and there would be root hair which would be present. This is microscopic presentation of a root hair. They are not as large as compared to the roots. So we are talking about this area, this is part of the soil, and this area is known as rhizosphere. Please remember, rhizo word is used for root, and we are talking about the area, the soil area which surrounds the soil, the root basically, and in the root here there would be few rhizobium which would be present, okay, in the soil particles. Now these rhizobium bacteria would form an infection thread inside this root, and they would reach till the cortex of the root. They have to reach basically. They would form the infection thread, they would come and accumulate near this root here like this. When they get accumulated, in response the root secretes lectins which brings more rhizobium close to it. So the next term that you come across is lectins. What are lectins? Lectins are the compounds which are secreted by the root of the leguminous plants, which brings more rhizobium close to it, and they get accumulated near the root here. When they get accumulated, in response to this accumulation, the root here would do some sort of curling. There would be a curling thing that would be occurring. Okay, as you can see, a curl has been formed. It is forming a hook-like structure around the rhizobium. Now this rhizobium would enter inside the root here and form the infection thread and reach till the cortex of the root. When it reaches the cortex of the root, the rhizobium, it forms bacteroids, small bacteroids inside the root's cortex itself. Now this bacteroid would be surrounded by a peribacteroid space. It itself is an entity, the bacteroid. There are many rhizobium which are present. This peribacteroid space, it separates the bacteroids from the cortex. All this is occurring inside the root cortex. Cortex is a region of the root where there are many parenchymatous cells, and it is in immediate contact with the vascular bundles of the cell. Okay. Now what are vascular bundles meant for? They are meant for transport of the various nutrients inside the plant. It would take the water and the nutrients to the top of the plant and would bring down all the food that has been prepared in the leaves to the roots and vice versa. So this peribacteroid space would be formed inside the cortex, and towards this peribacteroid space, what would happen? A component known as leg hemoglobin. Now this component of biological fixation is basically a compound, and you have to remember and learn it properly that it is an oxygen scavenger. Now you might be confused a bit that what are these terms doing over here? What are these bacteroids? What is this peribacteroid space? Why are we talking about oxygen scavenger and rest things? So please remember that the answer to all these things is that we have an enzyme known as nitrogenase. Okay. This nitrogenase is present in rhizobium. This nitrogenase is responsible for the reduction of this molecule of nitrogen to ammonia basically. This is what this nitrogenase does. It brings down the activation energy of this particular compound to such a level that it enables the reduction and addition of hydrogen to it, and ammonia is being formed. This is what it does basically. Okay, so it is nitrogenase which has to do this process. This nitrogenase is present in rhizobium, and this nitrogenase does not work in the presence of oxygen. For that purpose, the rhizobium forms bacteroids. It forms the peribacteroid space around which leg hemoglobin comes, and then the process of the reduction of nitrogen takes place. Okay. Now this reduction of nitrogen is exactly the biological fixation of nitrogen, where we would end up with two molecules of ammonia with expense of some sort of energy that would be in the form of ATP. Now what happens is that you have a nitrogen molecule. Over here 8 electrons would come into scene, 16 ATPs, okay, and they would convert this nitrogen into ammonia. These 8 electrons would be utilized for reduction process, and this is what is the entire biochemistry can be represented in this equation, how the biological nitrogen fixation has taken place. Alright, now this process is accompanied by the presence of nitrogenase. Now this nitrogenase enables, supposedly this this is an enzyme, this would enable this nitrogen molecules reduction by, why haven't I written hydrogen ions, sorry, this is what was missed. Of course there would be reduction, so it would need 8 hydrogen ions as well. So this is the entire equation. Please pardon me, I had not considered H positive over here. This nitrogenase would enable the addition of these H positive to nitrogen molecule, and this would give rise to ammonia. Now what happens next to this ammonia, which is formed as a result of biological nitrogen fixation, is studied under the fate of ammonia. In the fate of ammonia, we know that there is reductive amination which takes place. This ammonia gets associated to some amino acid like alpha ketoglutaric acid in the presence of an enzyme, and it forms the glutamic acid. So this is what is to take place inside the plant. The various biochemical reactions would take place, transamination would take place, there would be certain reactions which would be taking place. For that we needed ammonia, or we needed nitrate and nitrites, and this is how the nitrites and nitrides are available to the plant takes them up and then enables the assimilation. So in the beginning we had discussed that we should be able to understand these points. Now I hope you are quite clear with what are the available sources of nitrogen. One would be atmospheric nitrogen and the soil nitrogen, which is in the form of nitrates and nitrites. Then how that nitrogen which is taken up, that would be assimilated. What are the two types of fixation, out of which the biological fixation is very, very important, because maximum of the nitrogen which is available to the plants, that is available through the biological nitrogen fixation. The few prokaryotes which are involved in this, apart from that, we named 2 bacteria which were involved in physical uptake of nitrogen as well, physical fixation, and how the nodules are formed in leguminous plants and what happens inside the leguminous plants. One thing you need to remember is what is the function of leg hemoglobin, why the oxygen scavenger is required, and what is the role of this enzyme nitrogenase. This is all you have to understand about the nitrogen fixation. In the subsequent section we are going to understand nitrogen fixation pictorially so that you get an idea what actually is happening in the nature.